today I'm going to talk about my lab's work on modeling the basal ganglia to understand Parkinson's disease. So my lab, Laboratory for Computational Neuroscience at IIT Madras. So I head this lab, and the goal of this lab is to build a whole brain model. It won't be a detailed model like some of these larger projects, like what's happening in the Blue Brain project in Geneva. It's a simplified model with about maybe 10 million neurons instead of 100 billion neurons. And it will have representations of all the major structures in the brain. Once this kind of model is completed, I think it will have immense applications in both engineering and medicine. So this is like we call this model, like this hypothetical model, the mesobrain, which is cast at the mesoscale level. And so you can see that the ascending pathways, sensory pathways on the right side, the descending motor pathways, hierarchies. So you can see the ascending sensory pathways on the right and descending motor hierarchies on the left. And between the two, you have the prefrontal decision-making regions. And then there are three major loops: cerebellum, basal ganglia, and hippocampus. So if you can understand this kind of a cartoon picture of the brain, I think you have got it. You understand how mammalian brain works. I think rest is detailed. So I know I'm oversimplifying it, but even this is quite a challenging problem. So to this end, we have divided the lab into various teams. Each team works on one part of the brain and with a disease to bring that area into focus and give it an applied angle so that you can get some funding for that part of the research. So we have several teams in the lab. So one team working on vision, another on basal ganglia, another on hippocampus and navigation and neural oscillations, neurovascular coupling, and so on. So today's talk, I'll talk about our work on basal ganglia. So the goal of this team or this part of the lab is to understand basal ganglia in all its multifaceted aspects, and with that model understand various symptoms of Parkinson's disease, because that's what is affected, this circuit in the brain that is affected in Parkinson's. So this is a cartoon, like a block diagram of the basal ganglia. So basal ganglia, basically it's a subcortical circuit consisting of about 6 or 7 nuclei. This circuit receives inputs from the cortex and returns signals back to the cortex via the thalamus. And here you can see that the input port of basal ganglia is striatum, which again has 2 subregions. I'm not going to bore you with jargon, so I'll go easy on the jargon, right? And then there are 2 output ports, GPI and SNR. So there's a direct connection between striatum and the output, and therefore that is called the direct pathway. And then there's a longer route from striatum to GPI or SNR, and that goes via GPE and subthalamic nucleus and all that. Because yesterday there was talks on DBS, and SCN is an important target for DBS. So this longer pathway is called the indirect pathway. So everything then returns to cortex via thalamus. So basal ganglia is exciting because although it's a small circuit in terms of number of neurons or volume and all that, it's a very important and influential circuit because any damage to basal ganglia will have repercussions in all the 4 major domains of brain function: sensory motor function, cognitive function, affective and autonomic functions. So therefore, this is what you see in Parkinson's, for example, because of loss of cells in a tiny part of the basal ganglia called substantia nigra. So the British neurologist Marsden spoke of the mysterious motor functions of basal ganglia, and almost it's like describing some kind of a god, right? Because it's so mysterious and it's been bothering people for a long time. So people have been modeling various aspects of basal ganglia's functions. They separate models for reward-based learning, sequence generation, working memory. I mean, in fact, this list is a somewhat old list. There's constantly, every year, the number of papers on basal ganglia models is growing internationally. So, but the problem with basal ganglia models, and not only that, if you take any system in the brain, the problem is this kind of a piecemeal approach, right? Every group looks at brain from one angle, one tool, right, and talk about that from that viewpoint. And the big picture is lost. It's like this blind monks trying to figure out the elephant, right? One monk thinks it's like a pillar, another thinks it's a snake, and so on and so forth. So how do you get an integrated picture of basal ganglia? So and how do you achieve that using a comprehensive model? So now talking about Parkinson, it's a neurodegenerative disorder because of loss of cells in this substantia nigra. We don't fully understand why these cells die, so therefore we call it idiopathic. So we have a fancy way of clothing our ignorance in complicated Greco-Latin terms. The 4 cardinal symptoms of basal ganglia are tremor, rigidity, postural abnormality, and bradykinesia. And in motor domain alone, there are so many submodalities which are affected, which can be affected in general. A given patient may not have all the symptoms, but in general, you can see problems in reaching, handwriting, eye movements, gait and speech, and so on and so forth. So it's can be a pretty devastating, disease. Now, how does this disease work? I mean, that means how does basal ganglia work, right? Let us start with a classical picture. So if you take any textbook on neurology, or even if you take the Bible of neuroscience called Principles of Neural Science by Kandel, Schwartz, and Jessel, the story goes something like this. There are two pathways, direct pathway and indirect pathway. So if you activate the direct pathway, that facilitates movement, because output of basal ganglia goes via thalamus to motor cortex and other prefrontal areas. So it has a kind of it controls kind of the gateway to motor apparatus. So if the gate is open, movement occurs; gate is closed, movement is stopped. So that gate is controlled by these two pathways. If the direct pathway is affected, the gate is open. Therefore, it is also called the go pathway. And why does it happen like that? Because if you look at if you argue in terms of the neuromodulator or neurotransmitter stages, so from striatum to GPI, it is a inhibitory or a GABA pathway. GPI onwards to thalamus, it's a GABA again. So two negatives make it a positive. So that's the kind of arguments people propose. On the other side, if you look at indirect pathway, you have striatum to GPE is GABA, and GPE to STN is GABA, STN to GPI is glutamate, and beyond again you have GABA. So you have three negatives making a negative on the whole. So if you activate the indirect pathway, you have on the whole inhibition. So it's like one is pushing it, another is pulling it. So these two act together and cooperate and control movement. And what is even more interesting is, you see the projection coming from the SNC to striatum. So SNC releases a special chemical called dopamine, which is a key player in all basal ganglia functions. So this chemical, right, the levels of dopamine in striatum acts like a knob, like a switch, which controls the transmission of the corticostriatal inputs coming into striatum, right, and determine whether this information goes straight by the direct pathway or take a detour and go along the indirect pathway. So like imagine like a valve sitting there and controlling the flow and determining whether it goes straight or goes and takes a longer route. So therefore, if under high dopamine conditions, the direct pathway is selected, so you get to move. Low dopamine condition, that pathway is selected, so you stop and you have difficulty moving, right? And this is what happens in Parkinson's, because in Parkinson's the SNC cells are dying, and therefore there's lesser levels of dopamine. So information goes by the no-go pathway, and there's difficulty in movement. Now, this is a kind of a story that we were told in a standard textbook of neurology. Now, is this story correct? Let's take a closer look at this. So this kind of a simple binary description of basal ganglia's function anatomy, right, as something that's pushing and something that's pulling back, right, seems to be not very adequate. And to understand, and so there's a need for a broader theory, grander theory that explains all these many dimensions of basal ganglia. I think you need to take a closer look at what exactly the dopamine signal is saying, what is it doing. So for that, you need to understand this other aspect of dopamine, which is that it's called a pleasure chemical. Dopamine is brain's pleasure chemical. Whenever you're feeling happy, right, which means dopamine cells are singing. So this dopamine release is triggered by brain stimulation, psychomotor stimulants. When you take drugs, you go on high, right, that activates the reward system, activates dopamine cell activity in the brain. Opiates, and even food. You enjoy eating food because, right, you don't eat just for nourishment, you eat for enjoyment. That's because dopamine cells are firing. And dopamine antagonists, those chemicals which counteract the action of dopamine, also block the sense of pleasure that you get when dopamine is activated. And at a higher level, at cognitive level, even presentation of beautiful faces, images of lovers, even monetary rewards. When somebody suddenly puts a wad of cash in your hand, you feel very happy because dopamine cells get activated by that. So this aspect of dopamine is known for a long time. So, but way back in the late 90s, this group from Oxford, I think, Wolfram Schulz and his colleagues have done some interesting experiments that revealed an even subtler aspect of dopamine cell signaling, right, as it goes, as you can see in this slides. So this experiment is done on primates, right, under Pavlovian conditioning setup. The recording is done from ventral tegmental area, which is another dopamine cluster in the brain. So here the animal is given a conditioning stimulus like a flash of light. After a short delay, it is given a piece of apple as a reward. So here in this slide, in this trace, this is a dopamine cell firing in VTS in VTA. Conditioning stimulus is given here. After some time, the animal touches an apple. So you see that right moment it touches the apple, the firing increases, because apple means reward. So VTA cells, it means the signaling means reward, which is well known. So that's consistent with what we already know. Now look at the second trace, right? The same, you see the firing here, but in this case the condition stimulus is given. The firing began right after the condition stimulus CS is given, right? It doesn't even wait for the apple to come. Why is that? Which means that the cell's activity anticipates reward. It doesn't even have to wait for actual reward to come, because the CS and the reward are paid consistently. So the moment it sees that conditioning stimulus, a flash of light, it knows the reward is going to come. So cells are firing in anticipation of that. So this dopamine cell activity means not just reward, but actually anticipated reward or expected reward. That's what the second trace is saying. If you go to third trace, in the third trace again it's the same experiment, but with a small change. Right, the condition CS is given after a small gap when the reward is actually supposed to come, and they cheated the monkey. They removed the piece of apple. This box is empty. So monkey puts his hand in the box and find that the apple is missing. Right. So what do you see there? You see a small dip in dopamine cell activity. So monkey is kind of disappointed. Okay. So what this signal is saying is not just reward, not just expected reward, but the difference between expected reward and actual reward that it got. It's called reward prediction error. The error in reward prediction. Now, moment you see any brain signal which means some kind of an error, right? The machine learning and AI guys will get very excited, because the whole theory is constructed around some kind of error signals. You take an error signal and feed it into an error, gives you a way to train systems and networks. So this kind of error people have found. I can't go into all the details, but this kind of error people have noticed very early on. This 97 paper by Schulz and Reed Montague and Sejnowski. They found that it looks a lot like the reinforcement learning and a signal that appears in reinforcement learning theory, which is a branch of machine learning. So what is this reinforcement learning? So if you take an agent, right, where you give a stimulus to the agent and look at the response of the agent, if the response leads to a reward, then the agent will try to repeat that response to that stimulus next time, right? So that the stimulus response connection is reinforced. But if the response leads to punishment, the agent will try not to do that response to that stimulus next time, and the stimulus response relationship is attenuated. To give an example, let us say there's an animal in the wild, found some red moving thing, it just grabbed it and ate it, and happens to be a very juicy insect, right? Then next time it sees a red moving thing, it will grab it. But suppose that insect happens to be a poisonous insect and animal fell sick or something. Next time it sees a red moving thing, it will try to run away from it or avoid it. This is reinforcement learning, to put it simply. Now, is basal ganglia doing something like this? Just let's look at the anatomical context of basal ganglia. It's a subcortical circuit receiving input from both motor cortex and sensory cortex. So that's this big arrow you can see here. It's receiving reward-related information from SNC. So if you look at this box, three things are coming together in this box: stimulus, response, and reward or punishment information. Now, if you look at this box, basal ganglia, right, you get input from sensory cortex, which is telling you what is the stimulus, motor cortex, which is telling you what is the response, and SNC, which is telling you what is the outcome. Did you get reward or punishment? All three are coming together here. So you can right there see that basal ganglia is possibly a good candidate for performing reinforcement learning kind of functions. So that becomes a basis to understand, to create a comprehensive theory of basal ganglia. But then, so with that as starting point, people have developed lots of models of basal ganglia. So I'll tell you some problems with those models and what we have done to kind of correct that kind of a deficiency. Now let us go a little bit deeper into this. And how do animals or agents learn by reinforcement? Let's imagine a hypothetical situation where an animal is presented with two choices: pressing a yellow button or pressing a red button. If it presses yellow button, it gets some juice, which is reward. If it presses the red button, it gets a shock, which is punishment. But animal doesn't know that initially. It thinks both are equally good. So its weightage for both these choices are equal initially. And this weightage, I'll give a more formal term for it. So it presses the yellow button, it gets reward, so its weightage for yellow has gone up, right? It presses the red button, it gets a shock, so weightage for red has gone down. It does a few times, and yellow weightage maxes out, red weightage becomes almost zero. So these weightages are called values in reinforcement learning terminology. And these values are like surrogates to reward, because reward is something that you get after you perform an action, but that's too late, right? You want to know what you're going to get once you perform the action. So these weightages will guide you, in choosing the action. These weightages are called values. So if you want to have a reinforcement learning system, you need to figure out how to learn these values by trial and error, by performing actions. Okay, that's not the choosing of an action is like go in basal ganglia, and the not choosing of an action is like no go in basal ganglia. So you can see how the basal ganglia's functional anatomy can do these tasks. But there's a third thing which is missed in RL, the traditional approaches to RL applied to basal ganglia, which is exploration. What is exploration? Let me give an analogy. I mean, we all go to restaurants, right? Typically there are some our favorite restaurants in the city. We always tend to go to those restaurants because you get a good kind of bang for the buck. And then there are restaurants where you've been there before and you fell sick, and had to skip work and all that. So you you try to avoid those restaurants. But if that is your restaurant visit policy, that's not a very optimal policy, because there are new restaurants constantly cropping up in the city. You should be willing to try out new restaurants, and maybe they're the better restaurants than the best that you have known so far. So you should be willing to explore, right? As Nitish said, you should be willing to take risk, go out of your comfort zone and try out new things fearlessly. That is called exploration. So what is that exploration in basal ganglia? Nobody knows. Nobody talked about it, right, before. So if you want to think of basal ganglia as a comprehensive, a complete reinforcement learning engine, it needs to have a reward signal. It needs to have some way of representing values. It needs to be able to explore, try out new actions, right? It needs to perform actions. It needs to have a representation of the state. These are the 5 elements of reinforcement learning. So if basal ganglia is a reinforcement learning system, where are all these things happening in the circuits, right? So striatum, so state comes from the sensory cortex. State means, what is the state right now? That comes from sensory cortex. It's like stimulus. Now actions are motor actions, so that happens in the motor cortex, way downstream. The values, so there's a lot of data from neuroimaging, right, with human subjects performing gambling tasks, where people have shown that the part of striatum, the ventral striatum, is where the values are computed and stored. So I'll just take that piece of experimental information as it is. Okay, so but what about exploration? Where does exploration happen in basal ganglia? Right, so the classical view doesn't tell you anything about it. It doesn't even bring up the topic of exploration, because that's why I said go no go is not a complete story. And even this traditional story where you try to argue and support, justify go no go in terms of the neurotransmitter stages, it's not a complete argument, because the indirect pathway consisting of STN and GP, it's not a feed forward, it's not a bunch of feed forward stages. There's a feedback coming from STN to GPE that makes it a loop of excitatory inhibitory neuronal pools. And whenever you have a pool or a pair of excitatory inhibitory stages, you have very complex dynamics, right? So the simple arguments in terms of this is, how much time do you have? Okay. So we propose that the indirect pathway does not only no-go but also exploration, right? So we call this go, explore, no-go picture of basal ganglia, or gen model of basal ganglia. So how do we say that SNGP is exploration? So I can cite some more experimental evidence. So decorticated kittens in which cortex is completely removed, they're capable of showing exploratory behavior. So that means something is coming from the subcortex. Then STN lesion. So if you lesion STN, people show what is called perseverative behavior. What does it mean? They perform the same action even though that action is not giving them reward. That means they're not able to learn from reward. They're purely explore. They lost their ability to explore. And also people have found from electrophysiological studies, a huge number of studies like this, that the STN dynamics loses its dynamic complexity under conditions of Parkinson's. That the activity is less correlated or more desynchronized under normal conditions and more correlated in PD conditions. So with this background, I'll justify that STNGP system is indeed the explorer. This is what is driving your ability to choose actions randomly. So I'll describe a small model to start with, then I'll develop this. So I think I'll rush a little bit. So we developed this model here. In this model, this is a striatum which has 2 kinds of cells, D1 receptor expressing medium spinal neurons and D2 MSNs and so on. Right, and then the SNGP in the system shows oscillations. So I just want to say that this is not like an AI model where it's like a black box where you have garbage input, garbage output, and train on some data. It's not that kind of model. It's built from first principles, from a knowledge of basically anatomy and function and neurochemistry and all that. Okay, so these two combine in GPI and then output goes to thalamus. Okay, now when we give this network a very simple task, I have two choices, A and B. A is better than B, right? But which one will you choose? You should choose A, right? But what does the network do? Then we vary the dopamine level in the model. So when dopamine is high, it chooses A most of the time. When dopamine is low, it doesn't choose anything. That's like no-go, which is like consistent with what we already know. But when dopamine is intermediate, in the intermediate zone, it does something very strange. It keeps choosing B quite often within a probabilistic fashion. Now the point is, network is not a probabilistic model. It's completely a deterministic model. Why is it showing a probabilistic behavior as though it's, it's exploring? Why does it happen? That happens because of the oscillations in SCNGP. SNGP is a network of oscillators, only oscillators. When you couple them, they produce chaos, chaotic dynamics. And this chaos is what is giving the network this exploratory power, the ability to choose actions randomly. You see this more radically when you have lots of output options. So we then extended this to what is called the unknown bounded problem, a classical problem in reinforcement learning. So network can learn how to choose the best action when you have a bunch of actions based on the rewards that it gets. So then we extended this to continuous action domain, because a lot of things Basal does they happen in continuous domain. When you are walking, when you are moving your hands, all this happens in the continuous domain. So we extend the equations to that. So so this is a recent paper on, we applied this to a bunch of cognitive functions, working memory and all that, and which was done in collaboration with a group in UC San Diego, published in Scientific Reports just recently. So we've been working on Basal models for the last about 15 years, and we published this book, research monograph based on my lab's work. It's a comprehensive model of Basal. It explains a lot of motor functions, eye movements, gait, hand movements, handwriting, so full spectrum, and some decision making, working memory, some cognitive, sequence processing, and so on. And so I'll talk about reaching in this. We haven't touched affective, that is emotions and all that, and we haven't touched autonomic function. Why do you get constipation? All that we haven't done. Sleep regulation in Parkinson, we haven't touched that. Although we have done some separate work on sleep modeling. So reaching, let's look at reaching. So reaching is where you extend your hand and touch something, touch a target. So this is a model of reaching that we developed. And in this model, so these bursts are like the STN bursts in the beta domain, in the beta band. I think yesterday somebody talked about that. This is like a regular irregular spiking that you see in normal condition. This is like PD condition. So these are various parameters we set to distinguish between PD and normal conditions. So this is STN firing patterns in normal, and this is STN firing pattern in PD, and you see the spectrum differences, right? So now under PD condition, you also see tremor. So this is like a sharp peak, the tremor peak in the hand acceleration data. So you see the three kinds of movements that you get. This is healthy case where the hand is reaching a target, and the rigidity case. So you vary the parameters in one way, you get rigidity, the hand doesn't move, it's stuck kind of. And vary the parameters slightly differently, you get tremor. You see that small shaking of the hand. You see that here, and the shaking happens in the around 5 Hertz, right, which is close to the real frequency it's seen in patients. So with that, I think I will stop. This is again a big model we developed, and I can't go into the modeling details. I mean, even one part of this has 170 equations, and I cannot bore you with it. You'll go to sleep. But just have a simple animation just to give an idea of how it works. So this is a normal, say you see the tremor in this case, right? This is coming out of the model, and so if you give L-DOPA 100 mg, it slightly improves bradykinesia. If you give 200 mg, it's even better. Okay. Yeah, so right now we're trying to put this whole thing on a quantitative footing, where you take quantitative data from a patient, put the data into the model, then you have a kind of virtual patient. Then whatever therapeutics you want to plan, do it on the model first before giving to the patient. So I want to thank the 1970 reunion batch. They have given us a fund to start this Parkinson's therapeutics lab, and acknowledgements, a lot of labs to students in the lab who have done their PhDs on this, collaborators, sponsors. Thank you.
Long live India and France friendship.